This is Holy Cross mock, and it's for component 2, and it's mock number 4. And we're on to question 8. So we've got in Africa, Lake Nabugabob, and it's separated from Lake Victoria 1,000 years ago. So you can imagine, if I draw a little, there we are, okay. So we might have a lake there with fish in. Excellent biological diagram. And maybe thousands of years ago, the water level goes down to this level here, and you can imagine these fish here are now separated from these fish here. So there's a geographical separation between them. Cichlid fish of the genus Haplochromis. Haplo, that's a single colour chromis. I don't know. Name the phylum and class. Oh dear. Right, phylum. So it's going to be something pretty obvious. And we're told they're fish, and you might know a little bit about phyla, possibly not a lot. Phylum is chordata, or kind of vertebrates. Chordata, and the class fish, or pisces, or you could say fish there. You might not have known this, but don't let that throw you for the rest of the question. Explain why the fish from each lake can be described as different species for 2 marks. You're straight away going to say your definition of a species in the kind of negative. So if a species is able to interbreed to produce fertile offspring, these are saying they're different, so it has to be in the negative. So unable to interbreed to produce fertile offspring. And on most days that'll be enough to get you the 1 or 2 marks that you need. On this occasion they've been a little mean, but they have given you 4 lines, so that's a bit of a clue. Any reason they want you to sort of come up with a bit more than that. So why they might not be interbreeding if they were in the same place. So if we get a fish from here and a fish from there that is now separated, then why wouldn't they interbreed? Well, they might be different colour, or they might have some behavioural characteristic that makes them unable to interbreed, or might breed at a different time of year, that sort of thing. So, for example, behavioural difference, or features, appearance, that sort of thing. So a cheeky little second mark there. Suggest how analysis of DNA or proteins might be used to supply additional evidence that the fish have descended from the other lake. Well, if you were looking at the DNA, what would you be looking for? So for example, analysis of DNA, well, we're going to do genetic fingerprinting or base sequencing, something like that. So let's put genetic fingerprinting or base sequencing, something like that. Seeing as you know about both those techniques, you could also say immunological comparison here, but these are the 2 techniques that you know about. And what would you be looking for? The question is saying they've descended from these ancestors, so we're looking for similarities. Now, look for close similarities between the species. So, for example, similar base sequence, similar genetic fingerprint band pattern, that sort of thing. And then part D, 4 marks, so nearly half an essay, explaining if they split, how do you get a separate species? So you should recognise this as, well, it's the speciation story that I've met with salamanders and all sorts of different things. And I'm just going to list all the stuff, and there's going to be 4 marks in there somewhere. So here's a kind of summary of the process. And these are the lakes, and remember that the lakes, the water level might have changed and they become separate. So we're talking about geographical separation, a barrier, that sort of thing. Here's our barrier between the 2 lakes. Is that sympatric or allopatric? That's allopatric, different place. And then the populations can't interbreed. So populations can't interbreed because of the separation, not because they're separate species yet. They could interbreed if they could, grow legs and wander across the barrier, but they can't. So they're just not able to because they're not in the same place. And then you get mutations and changes. So different mutations. So different mutations in each population. So for example, fish over here in Lake Nabugabob might turn the fish green, whereas in Lake Victoria another different mutation might turn them red. So different mutations going on, and this might give them an advantage or a disadvantage, depending on the environmental conditions, and so different selection pressures and so on. So each lake has different conditions, and so different selection pressures, and you get sort of genetic drift. So you get a different gene pool, different allele frequencies and so on, different mutations in the different, in the 2 different separated lakes. So leads to different gene pools, or you might call that deans. And this is what's going to prevent them from breeding, because this now there's a difference in in behaviour or appearance and so on, and that's where we've come from with our unable to interbreed. So we're back to part B again now. So we've got our differences that are going to prevent interbreeding, and so we are now at a separate species. So from that we'd certainly expect 1 mark, at least a mark there, and all 4 of those. So certainly 6 out of 10, sort of a C grade.